All right, let's talk about something truly revolutionary. We've all heard about gene scissors, right? Usually in the context of medicine. But what if we could use that same technology to fight climate change? Yeah, you heard that right. We're going to explore how we can use this incredible tool to help plants literally pull more CO2 right out of the air. Let's get into it. So look, we all know we have to cut our carbon emissions. That's a given. But here's the tough question. Is that really enough? I mean, what about all the excess CO2 that's already up there in the atmosphere? The reality is just hitting the brakes on emissions isn't going to reverse the damage. It's like overflowing a bathtub. You can't just turn off the tap. You've got to start bailing out the water. We need to actively start cleaning up the mess. And that's exactly where this idea of carbon dioxide removal, or CDR, comes into play. Now, this is a totally different strategy. It's not about slowing down our emissions. It's about actively pulling CO2 out of the atmosphere and then locking it away for good. And what are the best machines for the job? Well, they're all around us. They're called plants. Today, we're going to see how gene editing, this incredible technology, can basically supercharge this natural process. Okay, so here's what we've got on deck. We'll start with the key tech and then jump into 3 amazing strategies: how to get plants to lock carbon deep underground, how to soup up the photosynthesis engine itself, and even how we can get soil microbes to join the fight. It's going to be fascinating. Let's do it. So, what's the secret sauce? What's the master key that unlocks all of this? Well, it's a technology I'm sure you've heard of: CRISPR. The easiest way to think about it is like a biological word processor for DNA. You know how you can search for a specific word in a document and then cut, copy, or paste? CRISPR does that for genes. It lets scientists find a very specific piece of genetic code and then precisely edit it. And it's that incredible precision that turns it into such a game-changing tool for climate. Okay, to really get why this is so huge, we have to look down at the ground, the soil right under our feet. You see, our planet's farmlands used to be this gigantic carbon bank account. But ever since modern farming kicked off, we've basically withdrawn a staggering 487 times gigatons of CO2 from that account. That is a mind-boggling number. But here's the thing. Where there's a loss, there's also an opportunity. What if we could start making deposits again? What if we could put all that carbon back where it came from? And that idea leads us straight into our first big strategy, which comes from the brilliant minds at the Salk Institute's Harnessing Plants initiative. Their whole concept is, well, it's just so elegant. They're basically saying, hey, instead of spending billions on giant, expensive factories to suck up carbon, why don't we just upgrade the best carbon capture machines that already exist all over the planet? And of course, they're talking about plants. And this slide just lays it out perfectly. Okay, on the left, you've got your standard crop plant, right? Shallow roots, does its thing. But look at the right. That is the Salk ideal plant. Using CRISPR, they are literally designing plants to grow these massive, deep root systems. But it's not just about making them bigger. Oh no, they're engineering these roots to be way, way better at storing carbon for a really long time. And the cherry on top, this also makes the plants way more resistant to drought. It's a total win-win. So what's the magic ingredient here? It's a substance called suberin. You can think of suberin as this natural, waxy, cork-like material that's already in plant roots. And here's the key part. It is incredibly tough. It just doesn't like to break down. So by using CRISPR to tell plants, hey, make more suberin, especially in major crops like wheat and rice, scientists are basically turning their root systems into these amazing natural carbon vaults. We're talking about locking away CO2 for hundreds of years. So let's break down how this carbon sponge actually works. It's a pretty straightforward 4-step process. Step 1, the plant just does what plants do best. It breathes in CO2 from the air. Simple enough. Step 2, it takes that carbon and sends it way down, deep into its new and improved root system. Step 3, down in the roots, that carbon gets converted into that super stable, tough-as-nails suberin. And then step 4, the final step. When the plant eventually dies, that super-impact root material just stays there, deep in the soil, locking that carbon away safely for centuries. And here's the best part. Honestly, this is the kicker. This isn't just a climate solution. It's a farming solution. Think about it. Deeper roots and more carbon means healthier, richer soil. That soil holds onto water way better. And what does that mean? It means the crops are tougher, more resistant to drought. And that is a benefit that gets farmers really excited. It makes adopting this technology a no-brainer. It is the definition of a powerful win-win. All right, for our next strategy, we're shifting gears a little. We've talked about where plants store carbon, but what if we could go one step further? What if we could actually improve the engine they use to capture it in the first place? Yeah, we are talking about giving a tune-up to the most fundamental process for life on this planet, photosynthesis itself. You know, the physicist Freeman Dyson said it best. He said, if we can control the way plants handle carbon, the fate of carbon in the atmosphere is in our hands. Just think about that for a second. The ability to fine-tune this process gives us, well, an unbelievable amount of influence over our own atmosphere. It's a huge responsibility for sure, but the potential is just off the charts. To pull this off, scientists are zooming in on one very specific, very important enzyme. Its name is rubisco. Now, rubisco is the absolute heart of photosynthesis. It's the little molecular machine that physically grabs a CO2 molecule out of the air and says, okay, you're with me now. But here's the problem, the big catch. For being so incredibly important, rubisco is, well, it's kind of lazy. It's famously slow and inefficient. It's the major bottleneck holding back the entire global carbon capture system. So the big question is, can we fix it? Can we give rubisco a kick in the pants? Well, some really groundbreaking research is happening right now. In one study, scientists used CRISPR to tweak 5 different genes that are responsible for building the rubisco enzyme in rice plants. Now get this, their first attempt actually made the enzyme less efficient. And you might think, oh, well, that's a failure. But no, it was a huge success. Why? Because it proved the most important thing of all, that we can engineer rubisco. We can change its properties. And that's the crucial first step. It opens the door to eventually supercharging the CO2 uptake of all the world's most important crops. Okay, for our third and final strategy, we're heading back underground. But this time, we're looking past the roots. We're diving into the unseen world of the soil itself, home to literally trillions of invisible helpers, the microbes. This idea is all about engineering an entire underground workforce to help us reach our climate goals. And it turns out this isn't just a story about CO2 anymore. There's another villain lurking in the soil. You see, modern farming depends on tons of nitrogen fertilizer. But when crops can't use all that nitrogen efficiently, some of the local microbes convert the leftovers into nitrous oxide, N2O. And this stuff is no joke. It's a greenhouse gas that is nearly 300 times more powerful at trapping heat than CO2. So check out this incredibly clever solution. Researchers used CRISPR to make a tiny edit to a single gene in rice, a gene called NRT1.1b that basically acts like the plant's nitrogen throttle. And the result was incredible. The edited plants became way, way more efficient at using nitrogen. They just didn't need as much fertilizer to grow big and strong. And less fertilizer means, you guessed it, a huge reduction in those nasty N2O emissions. It's another piece of the climate puzzle solved with a simple, elegant tweak. But it goes even deeper than that. This is what's so profound. By editing that one gene, you're not just changing the plant itself, you're actually changing its relationship with the entire microscopic ecosystem that lives around its roots. We're starting to figure out how to use gene editing as this incredibly precise lever, a way to fine-tune the complex dance between a plant and its soil microbiome, all for the benefit of the planet. So, let's step back and look at the big picture. We've just walked through 3 different, but totally connected, strategies in the fight against climate change. And the amazing thing is, they're all unlocked by the very same powerful tool, CRISPR. And this table really sums it all up nicely. First up, you've got long-term CO2 removal by beefing up crop roots with that suberin. Then you've got boosting the actual rate of CO2 capture by giving that rubisco enzyme an upgrade. And finally, you've got reducing those other nasty greenhouse gases, like N2O, simply by making plants better at using nitrogen. 3 different problems, 3 targeted solutions. And I think the most important thing to take away from all this is that these aren't just a bunch of separate, random ideas. This is a toolkit, a brand new toolkit for redesigning our planet's biology to help it heal itself. It's not about fighting nature. It's about working with it, giving it a boost, and accelerating the natural processes that can undo some of the damage that's been done. And all of this leads us to one final, and maybe the most important question of all. We are gaining the power to literally rewrite the code of life. That's unprecedented. So as we develop these mind-blowing tools to solve our biggest challenges, we have to ask ourselves, how do we use this power responsibly? How do we make sure it's wielded safely, ethically, and for the good of everyone? That's the real challenge. And that's the conversation we all need to be a part of.